Can somebody confirm you can hear me?
Confirming.
Thank you. All right, before we begin, are there any questions? Close that down. All right, not hearing anything, so let's begin. Let me blow that up a little. Share my screen. I've got all the labs graded now, both times, one first and second time. And I think I've got all the lectures graded up to the point we're at. So you're doing well. Keep up the good work. Any questions about the grading? All right, today is March 3rd. We are supposed to be starting chapter 13. We're not there yet. We're still on chapter 14. And let me inform you, you're going to have a quiz 5, this Wednesday. Quiz 5 is the narrowest quiz. It's only going to be talking about chapter 8 and lab 9. So it's only going to cover that material. Any question about anything? In the lab, there will be no lab today. However, I will be logging in for the first 15 minutes to answer any questions. Your unknown notes pages is going to close this Friday, March 6th, and the unknown project is due 11:59 p.m. March 7th on Saturday. If you request a test on March 6th, it must be a test that will incubate for less than 24 hours. So if you request a test on March 6th, it has to be a test that you can get the results in 24 hours. Otherwise, you will not get it in time. Let me mention the unknown notes pages need to be finished. If it's finished, I put on the bottom in the comments that the student has finished requesting, meaning requesting tests. If you have not uploaded your last results and talked about what your unknown number is, meaning what species it is, then you have not finished your unknown notes. In the unknown notes page, I inform you what needs to be on the unknown notes pages for full credit. Remember, the unknown notes pages will be graded and will count like a regular lab. Any question about any of that? All right. Let's move on to chapter 14. All right, it's not coming up where I was. Is that up there? Can you guys hear me fine? It's telling me not hearing anything. Turn up the volume. Let me,
Yes.
yeah, you're fine. I don't need to turn up the volume on my end. It used to be that it would tell you where you left off here, but I'm not getting that anymore. All right, I have that we had talked about the extent of host involvement, talking about a primary infection and a secondary infection. Let me go ahead and close that down and save that. And move on to the next slide. Oh, that's not the next slide. My computer is taking a long time today. And let's close that down. All right. In the severity or duration of a disease, there's two terms you should know: a latent disease is a disease where the patient has a period with no signs or symptoms. The pathogen is actually inactive, such as the shingles is a classic case where the child, or usually the child, comes down with an infection causing chickenpox. The child has an active infection and the virus is replicating, and then the virus stops replicating, the active infection stops, and the disease enters a latent period, a latent disease, where the patient has no signs or symptoms. The pathogen just sits around, not doing much, not causing any harm to the patient, while the pathogen is in the latent stage. But the pathogen can then come out of latency and then start replicating, causing disease. And this causes the disease shingles. So the first time a human gets infected with this virus, the varicella virus or whatever, it causes chickenpox. And then all other times it will be shingles. And the patient has the virus in between. It's just the virus is just going latent, not causing disease, and we call it latent disease. Any question about any of that? And then there's herd immunity. This is where many immune people are in the population. It's actually vital for the spread of infectious disease and why we have, what do they call it, like flu shots and vaccinations. If you had a population, let me blow this slide up, where the majority of the people were susceptible, not immune, and then a few people in the population come down with a contagious or communicable disease, the disease spreads. And it will likely spread to the majority of the people in the population. If there is no immunity and the people are mostly susceptible. On the other hand, if the majority of the population is immune to disease, meaning they've been vaccinated, and then a few people become disease carriers, the disease will only spread to people in close association with the disease people, like this individual there and that individual there. These individuals are protected by herd immunity because the disease has to go to that one and then to that one before it can go to that one. And these people prevent the spread of disease in the population. So disease does spread a little bit, but only in people who are in close association with the infected individual. And it's the whole point of immunization to stop the spread of disease. And that is by herd immunity. Now, some diseases, you need 95% of the population or higher immune before the disease won't spread. And a classic case of this is measles, where you need 95% of the population immune, or else the disease can spread. And that is why disease is actually spreading, measles, in the United States at the moment, especially in some southern states, Texas, and it's one of the Carolinas. I think it's South Carolina. The children in particular are not immune, 95% of them, and so the disease is continuing to spread. Any question about any of that? With COVID-19 in the old days, I imagine it's a little higher now, you needed 90% of the population to be immune or the disease would spread. The United States has never approached 90% of the population being immune to COVID-19. And like I said, my understanding is each variant of COVID-19 has been more likely to spread, spreads more easily. So it's probably climbed up very close to 95% now. meaning 95% of the population has to be immune, or the disease will spread. Any question about that? All right, let's talk about the patterns of disease. Normally, a specific sequence of events is associated with an infection by a microbe before you get subsequent disease manifestations. First of all, you have to have a reservoir to exist where the disease can come from. The pathogen must be transmitted to a susceptible host. The pathogen has to invade the host and then multiply within the host. Furthermore, the pathogen has to injure the host. If there's no injury of the pathogen to the host, then the host will have no signs or symptoms of disease. And then the illness will end when either the host immune system destroys the pathogen or the pathogen kills the host. Any question about any of that? Or I suppose when the host dies of its own, I guess that can be another possibility. There are predisposing factors that make the body more susceptible to, and therefore affect the occurrence of disease. For example, females have a shorter urethra than males, so they have a higher incidence of a urinary tract infection. There are inherited traits such as sickle cell anemia, or two sickle cell anemia genes, that can make the patient more susceptible to getting disease. So sickle cell anemia patients are more likely to come down with disease. There's another trait which isn't entirely inherited, it's also partly environmental, but diabetics are more likely to come down with disease. And diabetes is both inherited, partly inherited and partly environmental. Climate and weather, it's well known, can affect the outcome of disease or the occurrence of disease. Everybody knows colds and flus are more likely to spread in the wintertime than in the summer. With the flu, it was assumed initially it was just because everybody was indoors, everybody shut their windows, and so the air in the house recirculated, and so you were more likely to get the flu that way. That is true. So all of that is true, but it turns out the UV light is weaker in the winter than in the summer, and so the UV light from the sun is less likely to kill the virus. And then the temperature of the winter stabilizes the virus, and so when a virus gets on somebody's coat, it's going to survive much longer in winter temperatures than it would in summer temperatures. So the climate and weather can affect the occurrence of disease by a number of different factors. Fatigue is well known to affect the occurrence of disease. This is something that college students frequently are fatigued, and if you're fatigued, you're more likely to come down with an infection. It's well known that age can affect the occurrence of disease. Certain disease like COVID-19, that you get much greater risk of developing a serious disease the older you are. It's well known that lifestyle can affect the occurrence of disease. For example, smokers are much more likely to come down with a respiratory infection than non-smokers because smoking damages the mucous membranes. And then chemotherapy also affects the occurrence of disease. Chemotherapy tends to weaken the host immune system. It also can weaken the body's, mucosa, making it more likely the disease can get established in the host and then can either invade the host before the immune system reacts or the immune system's too weak to throw off the infection. Any questions about the predisposing factors? All right. The development of disease tends to follow a specific sequence. The sequence is similar for both acute and chronic diseases. First, there's an incubation period. The incubation period is the time from when the patient first becomes infected till the time the patient first has signs or symptoms of disease. So within the incubation period, the number of disease-carrying organisms is low, but they're increasing in number. So this is both the incidence of the infective agent and the the intensity of the disease signs and symptoms, meaning down here, very low signs and symptoms, up there, high signs and symptoms. Also, higher organisms up here and fewer organisms down there. So obviously, beginning, you have no organisms, and then you become infected, and you have the incidence period until you show your first signs or symptoms of disease. Then that's followed by the prodromal period when you have mild signs and symptoms. Like when you're first coming down with a sore throat, you have to say to yourself, gee, am I coming down with a sore throat? And you have to think about it. And you say, yeah, I am. I must be coming down with something. That's the prodromal period, mild signs or symptoms. It's followed by the period of illness, where the signs and symptoms of disease are at their worst, most severe. You don't need to think about whether you have a sore throat or not. You know you have a sore throat. That's followed by the period of decline, where the patient starts getting better. If a child or an individual has a fever, the period of decline starts when the fever breaks. In the period of decline, the number of disease-carrying organisms decreases and the signs or symptoms also decrease. That's followed by the period of convalescence when you still have the disease-carrying organism in you, but they've dropped now below a threshold level where the disease-carrying organisms no longer cause signs or symptoms. So you have the disease-carrying organisms, but you have no more signs or symptoms because their numbers drop below a certain threshold. Any questions about that? And then that would be followed by the absence of disease where you have no organisms in you any further. No questions about any of that. Well, here's a question for you. During what period of illness are you not communicable with a communicable disease? You want to have a guess? I'll give you extra credit if you get it right.
Wouldn't it depend on the illness itself?
Not usually, because all of them follow this um,
Trajectory?
development of disease. There's always an incubation period, always a period of illness, usually a
Hmm.
period of decline, and then a period of convalescence. You notice I didn't mention the prodromal period. That one can
Hmm.
depend on the disease. Like a human cold, to my knowledge, always has a prodromal period, but the flu doesn't always have a prodromal period. For example, you could be going along and you're in the incubation period, you have no signs or symptoms of disease. The next moment, it feels like somebody hit you with a sledgehammer. There is no prodromal period. You move directly from the incubation period to the period of illness.
I would say in general, then I'd be looking kind of towards the back end of the period of convalescence, usually 48 hours post, last acetaminophen dose or something like
Yeah,
that, but more
yeah,
so on that side.
that would be logical, but you're still communicable in the period of convalescence because you do have disease-carrying organisms. In fact, you are communicable at least in the mid to late incubation period. Maybe when you first become infected, until the disease-carrying organism starts replicating. You might not be infective when you have the disease-carrying organism in you, but it hasn't replicated yet. So that would be very, very early in the incubation period where you're not contagious. But late in the incubation period and in the prodromal period, for adults, for many common illnesses, you're actually most infective in the late in the incubation period and in the prodromal period. Why are adults this way? It's because their immune system has ramped up, and in the period of illness, our immune system is attacking the disease carrying organism, and so you're less communicable in the period of illness for many common illnesses like colds and flus, for example. You're more
That's
contagious in the incubation period, late in the incubation period, and in the prodromal period. This is for an adult, and I think it goes back to our caveman ancestry where the adult was actively trying to protect the adult's children. Children, surprisingly, are not this way. It's like they don't care if their disease hits their parents and their immune system does not ramp up and then make the disease less communicable in the period of illness. You
that's
don't need
really
to know that.
interesting.
Yeah, that is interesting. In the old days, like when I was a child, I was not allowed into the hospital to visit my brother who got in a well, it was a motorcycle bike accident. And they said, no, you're a child. You can't, under age 13, you can't come into the hospital. Nowadays, almost no hospital follows that period. The reason was because children, if they have a disease, they don't ramp up their immune system to make it less communicable. And nowadays they realize, especially children seeing their grandparents, it's beneficial to the grandparents to have the social interaction, so they allow children into the hospital. But when I was a child, they actually did not allow children into the hospital because of that reason. But to answer my question now, you're actually contagious at every period of incubation because you have the disease carrying organism in you at every period. It's only after the period of convalescence when you're not contagious, you don't have any disease carrying organisms in you. That was a trick question. I'll never ask you a trick question like that in the exam questions. All right, let's talk about spread of infection. A reservoir is a source of an infection. A reservoir can be living or non-living. There are humans which act as reservoir, and for many human specific diseases like AIDS and gonorrhea, humans are essentially the only reservoir. It is true that AIDS can hit a chimpanzee, but almost every case of AIDS happens from another human. Obviously, there's the first case, or the first two cases maybe, which came from an animal, and we're not entirely sure, probably from people eating the animal, not maybe cooking the animal, and it might have been a chimp. Um, and that's how AIDS first probably got into the human population. But generally, for many human diseases, humans are the main reservoir. And this can be also for carriers who have an inapparent subclinical or asymptomatic disease. Humans can also be reservoirs as when they're carrying a latent disease. They can't usually transmit the disease when it is in latency. So if you got chickenpox, you're not going to submit the disease while you're in latency. But once you come down with shingles, you can then spread the virus. Any question about human reservoirs? There are also animal reservoirs. Some zoonoses may be transmitted to humans. A zoonoses is an animal disease that can be transmitted to humans. A classic case of a zoonoses is rabies. Rabies cannot spread from people to people, assuming, I guess, you're not eating another person who has rabies or you're drinking their blood. Actually, rabies doesn't go into the blood, so you'd have to eat the person. Rabies does not spread human to human because rabies does not come down into the saliva of a human, so you can't get it from another bite of a human. It has to come from an animal, and that's because the virus can get into the saliva of many animals, not in humans, though. So my point is rabies is a classic zoonosis. There are other animal reservoirs, like certain flu cases, your cats and dogs can get them. Many flus they cannot, but certain of them they can, in which case they can then spread the disease. Like COVID-19 can come from both cats and dogs. It's much more likely to come from a person, but it can spread to cats and dogs. Any question about any of that?
Yeah, just talking about the spread of disease, I know that there's feline HIV. Have we ever seen that kind of cross species, or is that very specific just to cats?
As far as I know, it's only specific to cats. It's sexually transmitted, so obviously it's not going to be spread unless something like that were to happen. But I don't think that virus can actually grow in humans.
Interesting. Thank you.
Uh, I think it only grows in, as far as I know, domesticated cats, although it could spread in maybe zoological cats. I don't know about that. Um, all right, there's also non-living reservoirs. This is important for many human diseases like botulism and tetanus, but there are non-living reservoirs including soil, water, and lettuce, and many non-living reservoirs cause human disease, usually gastroenteritis when somebody eats contaminated lettuce. They're not cooking the lettuce, usually not you cooking the lettuce, in which case you can come down with the disease. Any question about any of that? All right, the transmission of disease. There are three principal routes by which a causative agent can be transmitted from the reservoir to a susceptible host. There's contact transmission, such as a handshake, vehicle transmission, such as by a glass, where the infected person spreads it by the glass and then to the healthy individual. And there's vector transmission. Vector transmission is by an animal. Any question about any of that? A vehicle transmission is by usually an object, vector by an animal. All right, there are three different types of contact transmission. There's direct person to person transmission, such as somebody sneezing on their hand and then touching another person's hand in a handshake. This requires close association between the infected individual and a susceptible host. Another example of direct person to person transmission would be kissing. There's also indirect contact transmission, where the disease carrying organism spreads from the patient or the reservoir to a susceptible host via a non living object. We have a term for a non living object. We call it a fomite, such as when somebody sneezes on their hands and then touches money, and then that person turns in the money, and then the teller hands you the money, and then you become infected by touching the money. So, contact with contaminated money or a baseball, two players are playing baseball. One person is sick, they touch the ball, the other person touches the ball, and then it spreads by the fomite. There's also droplet transmission, where transmission occurs less than 1 meter. I'm not going to quiz you on a droplet. I'm just going to talk briefly about it. Droplet transmission, shown here, is where somebody coughs or sneezes, and you're within 1 meter, right to about here. If you get infected by this, it's called droplet transmission. But if you're infected by this region here, it's not called droplet transmission. It's called airborne transmission, which isn't even a direct contact transmission. And I just think this is a little arbitrary where they're drawing the line. And then one is contact transmission, the other is a vehicle transmission. And so that's why I'm not going to quiz you on it, because it's the same cough or sneeze. It's just where you are in relationship to the person. Any question about any of that? Example of direct contact transmission to people high-fiving each other, and then indirect, the baseball. There's also three means of vehicle transmission: transmission by an inanimate reservoir, like a fomite. There can be food, water, body fluids. There's waterborne transmission, such as contamination by sewage. This doesn't happen in the first world very often. Very, very rarely would that happen in the first world, but it happens all the time in the third world countries. Waterborne transmission by sewage. And then there's foodborne transmission, where somebody is sick, they touch food, and then somebody else eats the food. And then there can be airborne transmission. And as I stated, that would be when you get it from a cough or sneeze and you're more than 1 meter away from the individual. Like I said, I won't test you on that. All right, any questions about vehicle transmission? Vector transmission is transmission carried by an animal. So the pathogen picks up the animal picks up the pathogen and then carries it to the individual. There's two ways that an animal can do this: mechanical transmission and biological transmission. Mechanical transmission is by passive transport of the disease by the animal, such in this case, the fly is landing on something, picking up the disease carrying organism on its feet. The fly then lands on the burger, carrying the disease carrying organism to the burger, and then the burger is consumed by an individual, picking up the disease carrying organism. Mechanical passive transport. I should mention that the most important group of vectors are arthropods, especially biting fleas, ticks, and mosquitoes, but there's other animals that can transmit disease, like this fly here doing it passively. There's also biological transmission, and this is when the pathogen reproduces inside the vector. For example, malaria infects a mosquito. The malaria actually reproduces inside the mosquito, and then the mosquito bites the patient, transmitting the malaria to the patient. So malaria is spread by a biological transmission, meaning a biting mosquito. Any question about any of that? All right, let's briefly talk about nosocomial infections, also called healthcare acquired infections. This is one of the most important topics you can learn from microbiology. Nosocomial infections are infections that result from a hospital or hospital-like stay, meaning the patient did not have the disease-carrying organism until they checked themselves into the hospital or hospital-like facility, and they picked up the disease-carrying organism from the hospital or the hospital-like environment. So it's a result of the hospital stay, and if they never checked themselves into the hospital, they would have never come down with a nosocomial infection. It's estimated that 5 to 15% of all hospital patients acquire a nosocomial infection. And this is each year. That's 2 million people in the United States each year. This is 20,000 deaths each year in the United States from a nosocomial infection. It used to be the eighth leading cause of death in the USA. It might now be the ninth because these numbers came before COVID-19 came out. And COVID-19 probably is one of the top eighth leading causes of death in the United States. So it might have knocked nosocomial infection down to 9, as well as nosocomial infections have actually decreased in the last 5 or 10 years. So instead of actually being 5 to 15%, it's now closer to 5% of all hospital patients. And we'll talk about that in just a minute. The CDC estimates that about 1 in 10 hospital patients gets a nosocomial infection in the United States each year. These numbers are from, the mid 2000, and that's why I said in the last 10 or 5 years, the numbers have actually decreased. So it's about 1 out of 20 patients now. There's a total cost to this. You don't need to know the numbers, but it's a staggering number. Nosocomial infections result from the interaction of 3 factors. First, there are microorganisms in the hospital environment. That's unavoidable. Hospitals are major reservoirs of sick people. Sick people often are sick because they have a microbe causing the illness. Some normal microbiota are also opportunistic, and so they're in the hospital environment. And then some nosocomial pathogens are antibiotic resistant, and that is especially a problem. When you check yourself in the hospital, you come down with a nosocomial infection, and then the clinician orders the standard treatment, and they discover it's resistant to that treatment. So microorganisms are present, and then the patient who checks themselves in the hospital probably did so for a reason, and so many of the patients in the hospital are compromised host, one whose resistance to infection is impaired by disease, therapy, or burns, such as broken skin or broken mucous membranes, compromises the host, making it more likely for a pathogen to get established. Burns, injections, ventilators, urinary catheters, the administration of anesthesia, all of these weaken the host's ability to resist the infection. Some patients also have a suppressed immune system, compromising the host. Certain drugs, like chemotherapy, weaken the immune system. Radiation therapy and burns can all weaken the immune system. Certain drugs and the burns also make the patient more likely to get infected just because their defenses, like the skin or the intact mucous membranes, are broken. So the patient is more likely to get infected if they get exposed to a nosocomial infection. And third, there's a chain of transmission in the hospital, leading to the patient coming down with the infection. The principal route of transmission is direct contact transmission, such as patient to patient or doctor to patient. Actually, in the 1950s and 60s, doctors frequently were transmitting nosocomial infection. Take a look at any old medical movie. The doctor proudly displayed the stethoscope around his neck, and then he would use the stethoscope to listen to the patient's heart, touching it against the patient's skin. And then when he was done, he would just flop it down on his chest. There was no cleaning of the stethoscope. And then he would go to the next patient and he could spread nosocomial infection. Also, in the 1950s, maybe the very early 60s, doctors didn't wash their hands in between patients, so they could be spreading nosocomial infectious agents. So, especially in special care units, the hospital staff have to engage in behaviors to prevent nosocomial infection, such as obviously hand washing, cleaning the stethoscope in between patients. There's also indirect contact transmission via fomite. I obviously mentioned the stethoscope, that would be via fomite, but there's also urinary and IV catheters. Contaminated surgical dressing can result in indirect contact transmission. And then there's vehicle airborne transmission, such as where the ventilation system spread the disease. It is a concern in the hospital that they really do not stop the transmission of flus and colds in a hospital, and it's just from the ventilation system. Any question about any of that? All right, let's move on. How do you control nosocomial infections? There are certain procedures that are generally implemented in the hospital to control nosocomial infections. First of all, aseptic technique. You've learned this in microbiology now. When you go and work in the hospital or hospital-like setting, always follow using aseptic technique. And then you'll reduce the chance of spreading a nosocomial disease. Use proper handling of contaminated materials. Frequent and thorough hand washing also reduces nosocomial infections. If frequent and thorough hand washing is used, nosocomial infections actually will be reduced by 50%. When you're done with the patient, you should wash your hands so you do not carry a nosocomial infective agent to the next patient. And obviously, wash your stethoscope after each use. You have education of the doctors, the staff, the patients, and the visitors. Nowadays in hospital, there's a sign, at least in the elevator, if not outside of every room, and usually in the elevator and outside of every room in the hospital, there's hand sanitizer saying sterilize your hands or sanitize your hand to prevent the spread of disease, nosocomial infections. And so now the hospitals have gotten very good at educating the staff. That happened in about the 1960s, the late 60s. The patients, that happened probably in the 1970s and 80s. And then in the 1990s, they started, maybe in the 1980s, started educating the visitors as well, putting hand sanitizer, at least in the elevators, if not outside every patient's room. There are many other things you can employ for a nosocomial infection. What you can do is take a look at the online chapter notes, and I discuss some of the things you can use to prevent the spread of nosocomial infections. Because many of you are going to work in a healthcare setting, you should prevent the control of nosocomial infections. Any question about any of that? And like I stated, because of the education of staff, patients, and visitors, we've actually reduced in the last 10 or maybe 5 years the spread of nosocomial infections in the United States by about 50% of them. So instead of 1 in 10 patients on average, it's now 1 in 20 patients getting a nosocomial infection. Any question about any of that? All right, some terms of epidemiology. Epidemiology is the study of where and when disease occurs and how diseases are transmitted in populations. The CDC collects and analyzes U.S. epidemiological information. Every state has a number of diseases which must be reported. Usually the doctors send them to the county health clinic, which in Clark County would be sent to the health facilities in Vancouver. And then that county sends it to the state, which is, I believe, Olympia in this state. In Oregon, it's in Salem. I used to live in Oregon, so I know that. And then the state health organism sends it to the CDC. The CDC then publishes it, the Morbidity and Mortality Weekly Report, MMWR, standing for Mortality Weekly Report, each week and you can find that at www.cdc.gov. Any question about any of that? All right, two terms in epidemiology: morbidity, the incidence of a specific notifiable disease; the morbidity rate, the number of people affected in relation to the total population in a given time period; the mortality, the deaths from a notifiable disease; the mortality rate, the number of deaths from a disease in relation to the population in a given time. Any question about any of that? Any questions about chapter 14? All right, let's end that and move on to chapter 13. Chapter 13, viruses, viroids, and prions. Know the goals. What's the capsid and envelope? What's a spike? What's a bacteriophage? What's the lytic life cycle, the lysogenic life cycle? What is a prophage? What is a provirus? What is a latent infection? What is a persistent infection? What is a prion? What is a viroid? 2. Understand the characteristics, the structure, and the classification of viruses. 3. Understand viral multiplication. And then 4. And lastly, understand what prions are and what viroids are. Any question about any of this? All right. The goal of a virus is simply to replicate. How a virus infects its host cell, takes over the machinery of the host cell, uses that machinery to hijack the cell and force it to make new viral copies, which then escape the virus. You watch that little movie just telling you about what I just told you. Any question about any of that? All right, some general characteristic of viruses. When a virus is not within a host cell, it is generally inert. It displays little or no metabolic activity, and it does not replicate. A virus requires a living host cell to multiply, so we can say that a virus is an obligate intracellular parasite. It must multiply within a living cell by using the host cell's enzymes, nucleic acid, amino acids, and other host cell constituents, meaning parts, to have the virus replicate. A virus has no ATP generating mechanism. Viruses simply use their host cell's ATP whenever they need ATP. Viruses contain either DNA or RNA as their innermost molecule, which is their genome. So their genome is either DNA or RNA. It's never both. Outside of that genome, the virus always is surrounded by a protein coat. And here we're seeing a protein coat called the capsid around the genome of the virus. And this part of the coat has been cut out so we can look inside the virus. Some viruses have an outer envelope made of lipids outside the protein coat. But not all viruses have an envelope. Any question about any of that? Some characteristics of viruses. The host range is the range of host cells a virus can infect. It's determined by specific host attachment sites, like cell wall, flagella, and other cellular factors necessary for viral multiplication. Specifically, let me blow this slide up.
Wait, can you repeat what the host range is again?
The host range is the range of cells that the virus can infect. That's determined by specific host attachment site called receptors. It's a molecule on the host cell that can bind to the virus. And it's also determined by the viral molecules on the virus called adhesions or ligands that bind to the host cell receptor, meaning adhesion is a molecule on the virus that binds to a receptor, which is a molecule on the host cell that binds to the virus. So they are adhesions and then receptors, they bind to each other. And that determines the host range, mainly. And if the host receptor is missing, the virus cannot bind to it. Most viruses are only infected by the cells of one or a few species, and generally, they're only most host cells are only a few cells of one or a few species. For example, HIV can only infect cells of two species. One of the chimpanzees, there's two species of chimpanzee, and humans. It cannot infect any other species because other species do not have a host cell receptor that'll bind to the ligands or adhesions on the HIV virus. So most viruses only infect the cells of one or a few species. And with humans, we have T cells, which can be infected. We have macrophages, which can be infected, brain cells, which can be infected. But most human cells, like your nose cells or your skin cells, cannot be infected, or a bone marrow cell, cannot be infected by HIV. Any question about any of that? There are some exceptions, like bird flu, which can infect humans and other mammals, and obviously birds. And obviously, it's affecting more than one or two species.
So in general, they mostly only infect like one or like a few different species, not multiple.
Most viruses only infect one or a few species. Equine encephalitis is an exception. It can infect many different mammals and humans. Some of them, I'm not sure about equine encephalitis, but another one would be similar, would be drawing a blank on the name of that one. No, I almost had it. It'll come to me. Moving on, I don't remember the name of it, but it's one of the encephalitises, which can hit like birds and spotted owl and humans. And might even infect insects because I think it spreads from insect to bird. But most viruses only infect cells and only a few cells of one or more species. There are some exceptions. Bird flu would be a good exception. It affects most mammals and many humans, obviously, and many species of birds.
Were you thinking of West Nile by any chance?
Yeah, that was it. West Nile
Okay.
virus is the one I was trying to think of. Can infect birds and most mammals. All right, thank you. Viruses, the relative size of it, it's about 10 times smaller than the prokaryote cell, which is about 10 times smaller than a eukaryote cell. Obviously, Viruses have a range of size they can be. The very smallest virus is, let me blow this slide up, around 20 nanometers in diameter. Nobody's ever heard of virus bacteriophage MS2, one of the smallest viruses known. But you've all heard about polio. Polio is one of the smallest viruses that you know of, 30 nanometers long. The very largest, longest virus is actually Ebola virus, which is close to 1 micrometer long. Oops. However, Ebola virus is not the largest virus when we're talking about viruses by their diameter. The largest virus we're going to talk about is megavirus, which approaches 440 nanometers. That's 0.44 micrometers. And this virus is slightly larger than small or the smallest prokaryote cell in diameter. Any question about any of that? Oh, by the way, those are different sizes of viruses. They can range quite a bit from poliovirus or MS2, which is even smaller than poliovirus, to megavirus shown over here, or Ebola virus, the longest virus and the biggest virus that we're going to talk about. Compared to E. coli, which is a little bit longer than most cells, most bacteria cells, and then an average red blood cell, for example, for the eukaryote cell. All right, viruses, a single mature complete virus particle. We have a name for it. It's called the virion. Usually the term virion is used when we're referring to the newly replicated viruses in a host cell. Those we call virions, and they'll be released by the host cell. A virion has a nucleic acid. This nucleic acid can be either DNA or RNA. So the genome is either DNA or RNA. It is never both. The genome can be double-stranded or single-stranded. Double-stranded DNA is normal. That's what our cell's DNA normally is. Single-strand DNA can also be the genome of a cell, a virus. And single-strand DNA is not unheard of whenever a cell is replicating its DNA or whenever it's transcribing its DNA. The DNA will be single-stranded. Double-stranded RNA is unheard of in our cells, but there are some viruses whose genome is double-stranded RNA. And then there's single-stranded RNA viral genomes. And if you have an RNA that's single-stranded, you need to state whether it's negative single-strand or positive single-strand RNA. So if it's a single-strand RNA, you need to state whether it's negative strand or positive single-strand RNA. Positive single-strand RNA has the codon AUG in it, and it can be read by the ribosome. The negative single-strand RNA is the complement to the positive single-strand RNA. It will not have the codon AUG, and the cell cannot read negative single-strand RNA and translate it into protein. Any question about any of that? All right, the genome can be linear or circular. Remember, our genomes have linear genome, linear DNA molecules, but a prokaryote tends to have a circular genome, circular chromosome, usually just one chromosome, which is in a circle. So some viruses can be linear, some can be circular, and some viruses further have their viral genome in many segments. Like influenza virus has more than one segment, or you could say in human cells, more than one chromosome. And this should be normal to you because humans have their genome in segments, and we call each segment a chromosome. Admittedly, the chromosome is DNA and protein, but our chromosomes are linear. Any question about any of that? All right. All viruses have a capsid. This is the protein coat surrounding the genome. Capsids are composed of capsomeres. Let me see if I can blow this up. See if I can blow it up further. That's better. So here's the capsid, the protein coat, and you can sort of see each capsomere here. Each one of these little circles is a capsomere. This capsid is made up of one capsomere, meaning one protein makes all of the protein coat of this virus. It's just repeating numerous times. And you could say it forms the actually, this is an octagon. Uh, Many viruses have more than one protein in their capsid. This one only has one protein in the capsid. If there's no envelope present on the virus, like this one, there's no envelope present, you call that a naked virus. If, on the other hand, the virus has an envelope, then we call it an enveloped or enveloped virus. So this virus has an envelope. You'll note that in addition to having the envelope, it has spikes on the envelope. So the envelope is mostly a lipid bilayer. I'll blow this up a little. Making the envelope, and you can actually see the envelope. Oop, too far. Right there in a bluish greenish color. That's the envelope. And there's the DNA inside the virus. There is the protein coat, the capsid, the dark thing inside the green. And then the green is the envelope. This has purple reddish protein spikes or glycoprotein spikes. We call these spikes and they're only found on envelope viruses. So if the virus doesn't have an envelope, you cannot find spikes. They're only found on envelope viruses, not naked viruses. The spike is an antigen because it is the molecules that are furthest out in the virus, and these are the molecules that the host tends to react with. And if it's trying to remove this virus, it'll actually make antibodies and T cells go out and find this cell and then kill it. And how it recognizes this as a foreign cell is because it has these spikes, which serve as an antigen, allowing the host to recognize that this cell is external to the cell, foreign to the cell. So the spikes serve as antigens, which the host will react to. The spikes also on this cell are the furthest molecules out on the cell, and so those tend to be the molecules which are the ligands or adhesions that bind to the host cell receptor. Any question about any of that? So here we're finding a helicovirus. You can see it very easily there. I don't see it on this one, but there's a helicovirus. And then there's, that's one type of virus. The second type of virus is the polyhedral virus, a dice with many different sides. That one looks maybe an octagon. 1, 2, 3, 4, 5, 6, 7. So at least 7, but usually 8. Portions to the capsid. And there's an actual photographic electron microscopic image of a polyhedral virus, and that's the artist's rendition. We'll talk a little bit about bacteriophage T4. It's a bacteria cell that infects E. coli. Here we're looking at T4 here and there. I don't see it here, but let me blow that up. Oh, that's another virus. It's a complex virus. And then this is an envelope virus. You can see the envelope right there. And like I said, there are 3 types of viruses. A helical virus, where the virus takes on the helical shape. A polyhedral virus, where the virus takes on a specific shape. And this one is a polygon, meaning it has multiple sides. Probably more than 8. 1, 2, 3, 4 at least. 5, 6, 7, 8. Probably 9, because it's going to have a place for the virus to get into and out of the cell. All righty. Oh, and then the third category of a virus. So we have helical viruses, polyhedral viruses, and then complex viruses. Complex virus can take on almost any shape. Here we're looking at a complex virus there, but this is a complex envelope virus there. Any question about any of that? We'll talk a lot about T4 in this lesson because this virus has been studied more than any other virus in the world. Why? Probably just because this is a virus that grows in E. coli. And then virologists have been studying it for many years. Even though they couldn't put on the lid properly, they did put the viral components into the shaker can, which then I use, and even with all that handling, the virus is fine. All right, any question about complex viruses? Polyhedral viruses or helical viruses? All right, let's talk about the general morphology. The morphology is the shape of a virus. Viruses may be classified on the basis of their capsid structure, giving shape to the virus. For example, this is a polyhedral virus, and so it would be a polyhedral classified with them. This is a helical virus, and it will be classified with the helical viruses. Why do we do classification on the virus genome end product rather than on the genome itself? All right, nobody's answering, so let me answer that. Let's see, we're here. What was the question I just asked?
I couldn't really hear it because you like tilted your face down and it was kind of hard to pick up the last part of it. Something about genome and the shape of the virus capsid.
Oh, why are we classifying viruses on the shape of the virus rather than classifying them on the sequence of their DNA or RNA? It's simply because viruses have a very small genome, and the virus also evolves very rapidly to the host it infects. And so we don't know the evolutionary history of viruses. We do not know the evolutionary history of viruses when we sequence the virus. And this is largely, like I said, simply because the virus has so few nucleotides. So when we classify viruses, we don't go higher really than an order. There is no phyla of viruses. We do classify viruses into a kingdom, but the kingdom is the kingdom the virus infects. So there are animal viruses, and they will infect an animal cell. And there are plant viruses, and they infect plant cells. And so we're classifying viruses into the kingdom they infect. It's not really the kingdom of a virus, it's the kingdom of cell the virus infects.
And what was the back?
Question was, why do we classify them on the shape of the virus? It's simply because we can't classify them, at least high up, on their sequence of DNA or RNA. We can only classify them like their family, their species, their genus. We can't go much above order of the virus. It's just because we don't have a large enough genome size to know the evolutionary history of viruses. Any question about any of that? All right. So I already mentioned that T4 is a virus that's studied more than any other virus, and that's largely just because it infected E. coli, and then virologists started studying it, and then they just kept studying it because it was one they'd studied, and then they realized, geez, we know more about this than any other virus. Let's study it even further. So T4 is the virus which has been studied the most. It belongs to the T-even family of viruses. T-even, T2, T4, T6. They all look like this, where they have a shape looking like the lunar lander of the Apollo mission. They have a head right here that has the DNA inside it, or the RNA, if it tends to be RNA. And it has a different capsid protein than the rest of the capsid. So it does have a different capsomer protein. This one has at least one, two, three, four, five, probably six different capsomers. And that gives the tail this portion of the virus and that portion of the virus. Tails are structures seen that viruses use to help them infect the cells, like many bacterial viruses have a tail because it's difficult infecting a bacteria cell. And so the tail helps the virus replicate by actually helping the virus infect the bacteria cell. The tail helps the virus infect the bacteria cell. On this portion of the virus right there and there, the bottom of the tail fiber, is the ligands or adhesions of the virus that bind to the host cell. And it's going to bind to a specific molecule in the host cell called the host cell receptor. In this virus, we have the tail fiber, we have the base plate and the pins, we have the tail sheath, and then the head to the virus, capsid, and all of them are part of the capsid. Protein coat of the virus surrounding the DNA. And yeah, the tail isn't really surrounding the DNA, but it's part of the capsid. When a virus infects an animal cell, the host immune can respond. It can stimulate the host to produce antibodies, which will bind to and react and neutralize the virus. The antibodies bind to and recognize the virus surface proteins, and these we call antigens. So an antigen is something that the host can recognize as being foreign to the host and then mount an attack against. And so when the immune system recognizes and infected with the virus and makes antibodies, the antibodies will actually bind to the viral antigens. So that's what an antigen is. Some viral proteins can change or mutate frequently. This allows different viral strains to arise, and therefore different viral strains can affect the same individual more than once. A very good example of a virus that changes or mutates frequently is the flu virus. And after about 3 years after getting the flu shot, the protection from the flu vaccine will drop very low, 30% or less. And that's because the flu virus changes all the time, and every year it's going to change and mutate. And after 3 years, it's changed enough that the flu shot will no longer be giving you any real protection. And that's why after 3 years, you must obviously get another flu shot simply by the frequent change of the mutation of the virus. Not all viruses change or mutate rapidly. Like, for example, the polio virus, its vaccine was made in the 1950s. There's two vaccines. I think one was 54 and the other 56 or 58. That's a long time ago. That's almost for the one that's 54. That's 50 plus 25 years, 26 years. So that's like 75 years ago. And that virus has not changed, mutated, so the vaccine is still used today. Which is a good thing, because otherwise we'd have to make a vaccine every year, like the flu shot. They redo it every year. Just because the flu changes or mutates frequently. What's another virus that changes rapidly? Where the
I
old
know that
COVID,
HIV is one.
not well, maybe. I'm not sure about HIV. Yeah, it does change. It actually changes inside the patient. So
Yeah, and we have
changes.
just dozens, I think, of subtypes right now, particularly under HIV-1.
Yes.
Yeah,
So
it's
you're
interesting.
right. That's another one. I was trying to come up with COVID-19. Why the original vaccine of COVID-19 is no longer given is because the COVID-19 has changed, and then the first vaccine of COVID-19 no longer provides worthwhile immunity. It might provide some immunity, but it would not prevent you from getting infected. Oh, excuse me.
Doesn't like COVID and Hep C rapidly change?
Yes, certain viruses do rapidly change. The flu is the best known example, where every year they make a new vaccine. COVID-19, they also make a new vaccine about every year. I'm not sure about HIV, how rapidly it changes, or the other virus. Would you say Hep C?
Hepatitis C. Yeah, they have to do like really extensive blood work for like half a year to even like do a treatment for it.
Mhm. Yeah, like I said, some viruses change rapidly, but many viruses do not change rapidly. And a good example is the polio virus, where we're still using the vaccines made in the 1950s. All right, let's talk about viral taxonomy. Viruses are grouped into families. The families are grouped into genus, the genus into species, the species into subspecies. Families can be grouped into, let's see, what's the next one? Um, so order can be grouped into order, but above that, we don't have any groupings. And the reason is we just don't know the evolutionary history of the virus, so we can't group them into anything higher than an order. And generally, family is what most viruses are grouped into. When we group a virus into a family, all viruses of that family will be called viridae, like the herpes virus of family. We call it herpes viridae. The genus name will be the herpes virus, or you simply just say virus, like the herpes simplex virus. That's actually the virus species, not the genus. And then the virus species, herpes simplex virus, that's the virus species. And then there's two subspecies designated by a number, herpes virus type 1, herpes
I think it's
type
a, I
2.
thought it,
Go on.
yeah, for oral and then genital, right? I thought it was a letter.
Yes,
Maybe it's a number.
it's a number. Herpes simplex virus type
One.
1 is the oral virus, which over 95% of the US population has. And then there's herpes simplex virus type 2, which is the genital virus. And I don't know the number of the percentage of the population that has that one, but it's not important. However, you can get herpes simplex type 1 as a genital infection, and you can get herpes simplex virus type 2 as an oral infection. So it isn't quite correct to classify it that way. It tends to be a genital infection, but it can spread orally. And the only difference between the subspecies is the number. So they're the same species. They infect the same genetic informations. They infect the same ecological niche, meaning humans. Same cells too, the oral and the genital cells. However, the subspecies can be very different. For example, the poliovirus type 1 causes the disease polio. The poliovirus type 2 is a virus that we use as a vaccine, and it's, let's see, the Sabin vaccine is simply poliovirus subspecies 2. And that tells you how different the subspecies can be. In one case, we're using one as a vaccine to prevent getting disease. The other subspecies causes the disease polio. Any question about family, genus, species, and subspecies? Oh, subspecies for humans would be something like the race of the human species, meaning there's whites and blacks. Those are subspecies. That would be what like a subspecies would be of a virus too. Any question about any of that? All right, so viruses are grouped by the type of nucleic acid they have, DNA, RNA, single strand DNA, double strand DNA. by their replication strategy, by their morphology. We already mentioned that one. That's the helical virus, the polyhedral virus, and the complex viruses. And then they're grouped into family, genus, viral species because we can know them by sequencing the DNA. But above order, we don't classify viruses because we just do not know what classification they belong to. It's largely because the virus is small, so we don't have a lot of nucleotides to base the classification on. And then viruses change their base pairs quickly so that the virus quickly becomes adapted to growing in their host cells. So I've already mentioned the virus family. We end it with viridae, so herpes viridae. For the genus, it would be the herpes simplex virus. So we just have virus for the genus. The common virus name would be the herpes simplex virus, either subspecies 1 or herpes simplex virus type 2. You don't need to know the human herpes virus. This was a newer name for herpes simplex virus, and it's not being used even by clinicians, so I'm not going to discuss it. Meaning we still call it herpes simplex virus. Viruses must be grown in order to study them, and you have to grow them in a living cell. Here, we're growing a bacteria virus in bacteria. Let me show this from this current slide. So here you see the white in this area. That's from a lawn of bacteria growing on the plate. Let me blow this up a little. Here we can see the color of the agar right there. And each of these is a plaque caused by a virus which replicated in one cell, caused the cell to lyse. What happens? The virus goes out of the cell. When the cell lyses, the neighboring cell infects those neighboring cells, which then replicates the virus, lysing the cell, which goes out to further neighboring cell, which results in a plaque of bacteria on the plate. Any question about any of that? So this, we put the lawn of bacteria on the plate, and then we put the suspension of the virus on the plate, spreading it around, incubate the plate, and then we see these plaques on the plate. I used to do this when I was an undergraduate or graduate student, a graduate student. Furthermore, you can notice a difference in the size. That one is probably caused by 3 plaques which fuse together, this one right here. So that's why that plaque is so large. But that one right there, as well as that one there, probably why these are large and this one there and that one there are small is simply because this one, the virus landed on the bacteria cell initially. So the virus began replicating initially. In the show here. Whereas that one, which is a small plaque there, did not land initially on the bacteria. Come on, paint, let's work. It's not opening. Oh, there it is. Geez, my computer's really slow. Here we have a bacteria cell. And here's the plate. And here we have the virus, which lands here. And what you needed to do is we needed the virus, the bacteria to replicate. And the bacteria had to replicate at least 4 times, maybe more, because there's going to be some over here too, and some over here. You needed this bacteria to replicate until it came out here and then touched the virus. And then now the virus has infected this cell and can replicate inside this cell. And where is it going to go? It's going to go to neighboring cells, which would be, let's say this one now replicates here. It'll then go out to the neighboring cell, infect these cells, causing the plaque to form. And my point is this plaque formed much after the plaque which happened with the virus landing directly on the bacteria. So this plaque could get started right away. This one, it took time for the plaque to even get started. Why that plaque is small and that plaque there is larger. Any question about any of that? All right. The viral genome only contains a few of the genes necessary for the virus to replicate, such as the capsid proteins in the protein code of the virus. That has to be in the viral genes. And then any other genes necessary for the virus to replicate if the host cell is not supplying that gene itself. Like oftentimes, the virus will not synthesize the DNA of the virus. It'll simply rely on the host cell's DNA polymerase to replicate the viral genome. So the virus is taking over the cell. We call that hijacking, taking over the cell's metabolic machinery and then using the host machinery to replicate the virus. And it will use the host cell ribosomes, the host cell DNA, the host cell amino acids, the host cell's ATP, some of the viral enzymes to replicate the virus. The basic mechanism of viral multiplication is similar in all viruses. We are way past our time, guys. You got to tell me when I do that.
Sorry, I didn't want to interrupt.
Yeah. All right. So let's end it here. And obviously, I guess I'm going to start the lab right away because it's already past time for the lab to start. But when we go past the time, let me know and I'll end the lesson there. So let me ask, are there any questions?
So, does this apply to all lytic bacterial?
Bacteriophages? Yes.
Yeah. Okay.
Yeah. The virus multiplies by infecting the cell, hijacking it, and then using the cell's machinery to replicate the virus. All right, any other questions? All right, I will see you in the lab in just, let's give it 3 minutes, and then I'll start the lab. There's nothing in the lab, I'm just gonna be available for questions.